out there in Zoom world who I can't see. It gives me great pleasure to introduce Matt Witkowski today.
Pretty good.
Is that close? So Matt is speaking to us today from NYU, or at least close to NYU. And he is originally from Australia, as you will soon notice. But he did his both undergraduate and PhD training at the Walter and Eliza Hall Institute, working with some very famous personalities in hematopoiesis and blood cancers, including Doug Hilton and Ross Dickens. So during his PhD and his brief postdoctoral training there, he is, I think, where he developed his interest in lymphoblastic leukemia. And that is a focus that you'll hear he's kept to this day. So a few years ago, he moved to NYU, where he started to work with Yannis Ifantis. And there he published some very important studies early on, applying single cell technologies to understand the complexity of the bone marrow microenvironment. And now I think what he'll talk to you about today brings this work to an understanding of how leukemia actually remodels and influences the bone marrow microenvironment, and how this is involved in the process of leukemogenesis. So without much more, I'll just say one more thing, and that is, again, for those joining late, that if you want to put questions into the chat, either you can just put your name and we'll call on you to ask your own question, or if you want me to read it, then you can just put the question, if it's simple, in the chat box. Okay, Matt, take it away.
Thanks, Patricia. And thanks everyone for attending this. Yes, as Patricia mentioned, I'm in the Ifantis lab at NYU School of Medicine. And today I'll present two stories that one of which is published, one that is not published, surrounding B-cell acute lymphoblastic leukemia, which is the most common form of cancer in children. And it's the result of transformed B-cell progenitors that are really blocked in differentiation in the bone marrow. And they accumulate genetic lesions, leading to their accumulation throughout the marrow, and then they'll disseminate around the body of an individual to impede normal organ function. And really today, I just want to focus on two key studies that we've been working on. And the first is trying to understand how the immune system may evolve with acute lymphoblastic leukemia in the bone marrow and peripheral blood of individuals. So we use single cell sequencing approaches, as well as many other approaches, to try to identify the immune components of ALL, and then take patient survival data that we're able to obtain from multiple institutes around the US, and look at the prognostic value of these immune populations in leukemic survival. Then we present some preclinical systems where we could actually interrogate the interaction between different immune populations and leukemic blasts. And in the second part of the talk today, I'll actually discuss some CRISPR-based screening studies that we've been doing to understand how the surface antigen CD19 is regulated in human B-cell leukemia. And actually, a lot of this work was supported or heavily inspired by discussions with Eric Kohler at Colorado. So I'll discuss what we found there. But just in the way of B-ALL itself, it's actually an extremely well characterized disease intrinsically. The blasts themselves have been sequenced at many levels, including germline mutations and somatic mutations. For example, work from Charles Mulligan and Jim Downing were able to demonstrate that recurrent mutations that impacted B-cell development are recurrently somatically deleted in B-ALL. And what this does is cause that block in differentiation that is a hallmark of this disease. Furthermore, work from Charles Mulligan once again and many other groups have shown that mutations such as Icarus and P53 are actually enriched following chemotherapy at disease relapse. So these are proposed to be drivers of chemoresistance in B-ALL. And then more recently, out of St. Jude, they were able to perform bulk RNA sequencing on around 2,000 individuals with B-cell acute lymphoblastic leukemia. And that's what I've shown here in this plot. Each individual dot is an individual patient, and you can see bulk RNA sequencing of the different subtypes of B-ALL in their cohorts at St. Jude, both children and adults. So the point of this is to say that we do know a lot about the intrinsic drivers of B-ALL. However, the extrinsic drivers of B-ALL survival are a little less understood at this stage. And indeed, there have been many studies that have tried to look at extrinsic drivers of leukemic blast survival in B-ALL. For example, work from St. Jude was able to show, and this is also work from Katia Georgopoulos at Boston Children's, was able to show that Icarus mutant B-cell leukemias are extremely adhesive. And this may be a critical property in their survival that is targetable and may also drive their chemo resistance, this enhanced adhesion. Furthermore, it's been shown previously that leukemic blasts utilize a vascular endothelial microenvironment via the CXCR4, CXCL12 axes as a chemoprotective niche. And this is something that's been exploited using CXCR4 agonists in the treatment of acute leukemias. But in the case of B-ALL, it's very poorly understood how the immune microenvironment actually co-evolves with the emerging leukemia and throughout chemotherapy. And this is particularly important now with the emergence of immune-based therapies such as CAR T cells and bispecific T cell engagers such as blinatumomab, which really rely on optimal T cell function for their killing function. And to do this, you really want to create a permissive microenvironment that would allow these cells to impart their tumor killing properties. So the first question we really asked in this study was, what is the composition of the leukemic microenvironment? And can we track the immune cells that co-occur with leukemic blasts throughout the course of therapy? So we aim to map this complete system, but there's some really major considerations that you have to take into account when studying B-ALL patient specimens. And that is, when you receive, for example, a bone marrow aspirate, the vast majority of cells that you find in these particular samples are B-cell blasts characterized by, for example, CD19 expression or CD20 expression. And I've just depicted that here in the schematic where the samples that we're analyzing are basically all blasts. And there's a minor fraction of what we deem to be the non-transformed leukemic microenvironment. So this is really important when you actually look at real data from a patient. So a healthy individual bone marrow, and these are mononuclear cells isolated from a patient. Around 13 to 15% of the cells in this case are B cells expressing CD19. However, in the ALL patient here, you see an overwhelming number of blasts, which means that if you're trying to analyze by bulk RNA-seq and you just mash this sample up, extract RNA, the majority of signal you're going to get is from blasts. But there is this small population of CD19 negative cells that we don't really know a lot about. So we asked the question, what are these CD19 negative hematopoietic cells that coexist with leukemia? And to do this, we took a rather novel strategy at the time, which was to take these frozen bone marrow mononuclear cells from patient samples that we collected from all across the US. This included NYU, St. Jude, and the Children's Oncology Group. And it must be noted again that these are bone marrow mononuclear cells, so we're not getting information on granulocytic cells in these samples. And when you gate on these cells based on CD19, we actually use cell sorting to separate the CD19 positive B cell cluster and the CD19 negative CD45 positive immune cells that we assumed are enriched for the immune microenvironment. And then what we did here was we actually mixed these all back together at an artificial ratio where we had 5 parts of the CD19 population as the dominant group of cells analyzed, and then 1 part CD19 cells. And we used the 10x genomics version 2 chemistry of single cell RNA-seq, and this is just 3' single cell RNA-seq in this case. We compared these samples to healthy bone marrow controls, and the first question we really sought to ask in this analysis is what does leukemic cells do to the microenvironment when they emerge in the bone marrow? How do they actually remodel the bone marrow microenvironment initially prior to seeing any chemotherapy? So the comparison that we make here is the comparison of healthy bone marrow and diagnosis bone marrow in this case. And that's what I'm showing here on the top left. This is a UMAP plot, which is a 2-dimensional representation of single cell RNA-seq data. Each single dot here represents a single transcriptome from either healthy in green or blue and purple BALL-derived samples. These samples were integrated using the Seurat integration tool, which I can go into later on if anyone is interested. And this was performed to minimize the technical variance that exists between samples when analyzing single-cell RNA-seq from different sources. So we were able here to map around 50,000 single-cell RNA transcriptomes from healthy and diagnosis bone marrow cells. And we wanted to ask the question first, quite simply, can we identify the cell types that you would expect to see in a human bone marrow using a gene set enrichment analysis approach? So to achieve that, we actually broke these samples down into 96 independent clusters. And then we had 6 manually curated cell type specific gene lists that we applied to each cluster and assigned an identity. And as you can see down here, for example, these orange cells are our B cell compartment, and they are distinct from the purple myeloid compartment and appear to be branching off this hematopoietic stem and progenitor cell group. So it did recapitulate the hematopoiesis we would expect in a human individual. Then we have here a heat map just showing key genes that distinguish the cell types across the bone marrow. But the question was, what are the differences in representation of immune cell populations at the emergence of leukemia? And that's shown here in this heat map representation, where we basically plotted the density of cells from either diagnosis samples or healthy samples and looked at where the major differences occurred in hematopoiesis. So if we focus here on the B cell lineage first and foremost, you can see this accumulation of the diagnosis enriched samples. And this is actually that block in B cell development that we expect in BALL. However, when you just focus on the non-B cell fraction now and quantify the representation of cell types between healthy and diagnosis, we actually saw a very reproducible reduction in the number of diagnosis-associated myeloid cells when compared to healthy. What this suggested to us is that myeloid cells are particularly sensitive to the emergence of ALL in the bone marrow of individuals. So we wanted to explore this phenomena a little further. And what we did was we actually isolated the single myeloid cells from healthy and diagnosis individuals and replotted these cells. And when we did this, we're actually able to identify 6 major subtypes of myeloid cells present in the human bone marrow. And that's shown on the left using this heat map representation, or on the right here with these independent myeloid clusters found in these individuals. Now, despite having no granulocytic information, these cells predominantly represented monocytic and dendritic cell mature and progenitor cells. So then what we were able to do subsequent to this was look at the actual representation of either myeloid subgroups across either healthy or diagnosis patient samples, suggesting how differentiation might be impaired by the presence of BALL. And what we noticed is if you looked at diagnosis patients, there was actually this major enrichment for this healthy and diagnosis cluster 5, which is actually found at the apex of this UMAP plot. From this particular population, what we noticed in terms of transcript was that these cells had particularly low CD14 expression, but had high expression of mRNAs encoding CD16 and the MCSF receptor. So that's just shown here, and this sort of pointed us toward the idea. Sorry, is there someone speaking just now? Oh, no. Oh, we got it? I think. Okay, sorry, I'll keep going. So these are MCSF receptor high CD16-like monocytes that we believe to be found enriched in leukemic bone marrow. We then looked into the peripheral blood of leukemic patients, comparing healthy and diagnosis peripheral blood. And here I'm just showing the leukemia-associated myeloid cells. And once again, although we only had 2 patients per condition here, we saw that this group, cluster 2, shown in orange out here, was characterized by an over-enrichment in leukemic samples and was shown to be low in CD14 mRNA expression and high in MCSF receptor and CD16 expression. And the MCSF receptor expression is something to keep in mind later on. Now, one of the major advantages of this study was actually the fact that we had patient samples from diagnosis, remission, and relapse. And that was a major advantage for us, as we could actually track the kinetics of the immune system as the patient went into a loss of their bulk tumor population, as shown here, from diagnosis, CD19, CD10 positive blasts, into this now deplete fraction of that population following induction chemotherapy. And then around 3 years later, this individual relapsed with a CD19 positive, CD10 positive disease. And we could track the environment throughout all stages of disease using the approach I described earlier. And by doing this, we actually were able to sequence around 100,000 cells from 7 individuals with matched diagnosis and relapse samples, 6 of which we also had a match remission sample. And here, once again, we just identified our very basic immune cell populations at diagnosis, remission, and relapse. And along the bottom, I'm just showing some key markers of either B cells here with CD19 transcript, CD68 transcript for our myeloid cells, and then so forth for other cell types. But because of the dramatic remodeling that we observed in the healthy and diagnosis comparison with myeloid cells, we decided we wanted to really zone in on this population and understand how is the myeloid compartment changing throughout therapy when you remove the bulk leukemic population from the marrow and then when the tumor reemerges. So once again, we isolate our myeloid cells from this diagnosis, remission, and relapse analysis, or DRR here. And then we were able to show our 6 individual cell types present in the marrow of these individuals. And I just want you to focus here on this blue population, which is the myeloid cluster 1, which is characterized by high MCSF receptor expression, high CD16 mRNA expression, and low CD14 expression. And what we noticed in this population is that in stages of disease, diagnosis, and relapse, there was a significantly higher proportion of these cells when compared to remission samples, where we observed that there was actually very few of these cell types present in the myeloid compartment of these particular individuals. And based on the transcripts that we were observing, we started to think, well, these do appear to look like non-classical CD16 positive monocytes, but we wanted to validate that through a series of different methods, one of which was to use a previous human single-cell RNA-seq study from Rahul Satija and others in New York, where they were actually able to show here that when you isolate different human monocyte or dendritic cell subtypes, they created transcriptomes of each cell type. And mono2, for example, was how they labeled this CD16 positive monocytic group. So we compared our data set of this myeloid cluster 1 to the published data set. And what we found here was that our cluster was most transcriptionally concordant with their CD16 positive monocyte-like cells. Not only did we perform GSEA to validate the identity of cells, but we also performed a diffusion mapping, which gives somewhat of an understanding of the differentiation relationship between single cells in 2-dimensional space. Here we saw that the CD16 positive monocytes appeared to be the progeny of CD14 positive and CD14 positive, CD16 positive intermediate monocyte-like cells. And this is well known in the literature. The majority of non-classical monocytes do come from a classical monocyte source. And this transition is believed to occur through classical monocytes interacting with notch ligands present on the vascular endothelium, leading to the conversion into these patrolling CD16 positive non-classical monocytes. And that is work from Ralph Adams in Germany. We also used a recent single-cell RNA-seq technique known as site-seq. Here, not only do you get information on the transcriptome of a single cell, but you actually paint your single cells with surface antibodies that carry an oligonucleotide tag. So when you sequence the single cell transcriptome, you also sequence for the abundance of oligo tags corresponding to a given surface protein that you're analyzing. So for example, when we're looking at these additional patient samples and the myeloid cells we sequenced from these trios of diagnosis, remission, and relapse independent of our discovery cohort, if we focus in on this blue population here, we see that CD16 mRNA by the Fc gamma receptor 3A mRNA encoding a subunit of CD16 corresponds to high expression of the protein CD16 on the surface. So we were able to make the conclusion that the RNA reads that we were defining our monocyte subsets by did correlate with the protein reads. And then we also use flow cytometry on multiple validation samples where we're able to take either healthy, diagnosis, or relapse marrow and peripheral blood, and then perform flow cytometry on these samples. So I'm showing here the bone marrow of a healthy individual and a leukemic individual gated on NK cell deplete, T cell, B cell deplete cells by flow cytometry. And what we find here is in a healthy individual, the majority of these monocyte-like cells are CD14 positive, 16 dim, with a smaller fraction of our CD16 positive, 14 dim population. And we quantify that in the bone marrow and peripheral blood. The left column is classical monocytes, the right column non-classical. And in leukemic samples, now we get a complete switch toward the non-classical monocytes, validating our finding that the leukemic myeloid microenvironment is predominantly non-classical monocytes in the mononuclear fraction. So all this talk of non-classical monocytes is all well and good to identify them, but what do they actually do and how might they relate to the pathogenesis of this disease? So I'm just going to show a video here using a CX3CR1 reporter, which marks monocytic populations, as well as some T cell and NK cell populations, as well as dextrin marking the vascular endothelial cells. And what you'll notice is these beautiful little monocytes that they actually crawl along the vascular endothelial cells. And their role is to actually detect signs of inflammation or infection in the vascular endothelium. And upon interacting with damaged vascular endothelial cells, they trigger a TLR7 signaling cascade, which leads to the recruitment of granulocytes to the site of vascular damage, and then clearance of these damaged vascular endothelial cells. And due to their function in maintaining vascular integrity, these cells have been implicated in many inflammatory diseases, including atherosclerosis, MS, and rheumatoid arthritis. But their function in ALL is still very poorly understood. And in our own data set, one thing we noticed is if you isolated monocytes from these individual human patients and then compared the transcriptome of healthy or diagnosis non-classical monocytes, we saw that in leukemia-associated non-classical monocytes, we had an upregulation in genes such as CX3CR1 and PCAM1, known to be important in the patrolling and migration around the vascular endothelium of these cells. This was confirmed using our site-seq technology, where we see, for example, that the mRNA encoding, for example, PCAM1, corresponds to high levels of protein of PCAM1 on the surface of these cells. So this raises the question, how do BALL cells actually interact with these monocytes? Everything so far is quite correlative. So could we generate a platform where we could actually interrogate the interaction between BALL and monocytes in a human setting? So to do this, with my mate Chow downtown, he works at NYU Tandon School of Engineering, and he's a bioengineer. And what we wanted to come up with was a mimetic of the human bone marrow, where we could actually generate human vascular endothelial cells and infuse BALL into this vascularized microenvironment. So this schematic shows this circular structure where we have a central sinus seeded with human umbilical vein endothelial cells. And after about 10 days, they form vascular endothelial cells around this device in this inner ring. Now, what you can do in these particular devices is after you get your vascular endothelial cells, you can actually infuse human BALL cell lines into the central sinus of these devices, and they occupy the lumen of the microvessels that you can see here. So they actually can interact with the vascular niche. So we wanted to use this vascularized human tool, these are all human cells, to explore the interaction between monocytes and BALL. So the design of this experiment was to generate our microvessels with HUVEX and either leave the devices unseeded with BALL or infuse BALL cell lines. And we used either REH or SUP-B15 here. We allowed leukemic blasts to interact with their vascular microenvironment for 48 hours. And then we infused into the central sinus of these particular devices human classical monocytes from the peripheral blood of healthy donors. So these are CD14 positive, CD16 low human classical monocytes. And we allowed all these three cell types, the endothelium, BALL, and monocytes, to interact for 3 days, and then looked at what emerged in terms of differentiation of monocytes in this case. So if we just infuse monocytes alone, we do see after 3 days, CD14 positive and some expression of CD16 on the surface of these monocytes that are found in the vascular endothelial cells. However, when you preceded these devices with monocytes, now we can see that these CD14 monocytes are really turning on the expression of CD16 in the cell type, suggesting that there may be some mechanism by which ALL is promoting the differentiation of classical monocytes into non-classical. And we still require further mechanistic studies to understand this transition. We quantify that phenomena here, and I just want to make note that infusion of ALL alone, shown by this bar and this bar, actually increases the diameter of the microvessels that we generate. And if you infuse monocytes subsequent to that, you can actually rescue this change in vascular diameter, suggesting here that the monocytes may be playing a role in maintaining the vascular integrity that is compromised by leukemic blasts. But once again, we need to do far more mechanistic studies to look into this. The next question we want to ask is, are these of any prognostic significance, these leukemia-associated monocytes? And very few studies have analyzed the impact of the immune system on patient outcome under conventional chemotherapy. Often we're exploring cell intrinsic gene expression pathways and how they would drive therapeutic resistance. But here we were trying to look at a cell extrinsic influence. So the first study that we conducted was with a collaboration with Texas Children's Hospital and Karen Rabin, where she had pediatric BALL case peripheral blood data, where they collected the absolute monocyte count at disease diagnosis. And then they were able to actually stratify patients into absolute monocyte count high at disease diagnosis versus low, and track patient survival. And what you can appreciate are patients that present with a high absolute monocyte count in the peripheral blood have an inferior overall and relapse-free survival to those of low absolute monocyte count. We then wanted to explore a higher risk subtype of ALL, which is adult BALL. And although we couldn't find peripheral blood data on these in terms of absolute monocyte counts, St. Jude's bulk RNA-seq presented quite a unique opportunity, where what they've done here is they've not sorted for BALL blasts. So these transcriptional profiles they generate do harbor transcripts from leukemia-associated cell populations. So using a tool known as CyberSort, you can take existing gene expression signature matrices and apply it to bulk transcriptome information to predict the relative abundance of different immune cell types that may be present in these preparations. So in all of the adult BALL patients, we predicted whether patients would be monocyte high or low, and then looked at patient outcome. And whilst the subtypes of BALL were completely balanced between these two groups, monocyte abundance did predict an inferior overall survival in adults. And this was work with Charles Mulligan and Catherine Roberts. So the final question of this study was, we see an association between monocyte abundance and patient outcome and monocyte differentiation and BALL presence, but can we target these cells in vivo? So we turned to a model, a preclinical model of BALL, where I was able to drive BALL using a BCR-ABL P190 retroviral construct, which is tagged with GFP. And this preferentially drives B-cell leukemia in reconstituted C57 black 6 mice. I can then take BALL cells from these primary recipients and passage them through immunocompetent, non-irradiated black 6 mice, generating tumors that don't require irradiation for engraftment. We can see here that GFP positive cells transit to the bone marrow of individual recipient mice, homing to the vascular endothelium in red with the B cadherin reporter, as well as some potential interactions with CD68 positive myeloid cells. We performed extensive flow cytometry and single cell RNA-seq on healthy littermates and leukemic bearing mice. And the single cell RNA-seq is shown below here. And I just want you to focus in on these brown population of cells, which are monocyte dendritic cell-like cells. And when you look at this population, we used a marker known as TREML4, which is actually a really nice transcript to look at non-classical monocytes. And what we found was that in healthy bone marrow and peripheral blood, we saw that these populations were quite small in these organs. However, when mice were transplanted with BALL, we now see that there's a significant increase in the proportion of TREML4 expressing cells by single cell RNA-seq. By flow cytometry, if we gate on CD11b positive myeloid cells from mice, and on the x-axis we have monocyte chemokine receptor CX3CL1, we can see here that in a healthy peripheral blood, we have Ly6C high classical monocytes and a smaller proportion of Ly6C low monocytes here. However, if leukemia is around, now this shifts completely, and we see now a push from these Ly6C high cells down to these Ly6C low non-classical monocyte-like cells, consistent with our human finding that I showed earlier. Also, we are, similar to the human studies, our monocyte population expresses the MCSF receptor. We know this is expressed in other progenitor and monocyte populations, but we thought let's take quite a dirty approach, use an MCSF receptor blockade to deplete leukemia-associated monocytes and see if it changes the course of disease in vivo. So what we found is when you transplant BALL into immunocompetent Black6 recipient mice, and then you start treating with either IgG or this anti-CSF1R antibody, and this is the AFS98 monoclonal antibody for CSF1R. So it's competing with the MCSF ligand. In this case, when you deplete monocytes, and it is depleting classical, non-classical monocytes in this case, not just our non-classical. It doesn't actually change the course of disease at all by depleting monocytes. And this is something that's been observed in solid tumor studies by Lisa Cousins and others. It really requires a second hit where you start to kill the tumor at the same time to see the true effects of monocyte depletion on tumor-bearing survival. So here what we noticed is if you treat our IgG treated mice with nilotinib, a tyrosine kinase inhibitor that will target BCR-ABL, we can significantly increase the survival of these recipient mice, but around 85% will succumb to disease. However, if we deplete monocytes concurrently with the nilotinib treatment, now only 50% of mice will succumb to disease, suggesting here that leukemia-associated monocytes may actually play a chemoprotective role in disease. So for part one of this talk, we showed that BALL does alter the myeloid compartment of both humans and mice. The abundance of leukemia-associated monocytes predicts patient outcome, and that BALL itself does seem to drive the emergence of non-classical monocytes through mechanisms that are unclear. We showed that monocyte depletion enhances TKI sensitivity in vivo, but really there's a lot of questions that remain, having identified this association between these two cell types. We really don't know the mechanism by which non-classical monocytes promote leukemic survival in vivo or in humans. My hypothesis is it's really capitalizing on the normal function of non-classical monocytes to maintain the integrity of damaged vascular endothelial cells. And these vascular endothelial cells actually provide a chemoprotective niche for those residual leukemic blasts to reemerge. So what we want to do now is use live 2-photon imaging to visualize the vasculature, monocytes, and BALL interactions in vivo. Then we want to actually use a far more specific depletion of non-classical monocytes than the anti-CSF1R approach that we've used currently. And Lynn Hedrick at La Jolla generated a NR4A1 or NR77 enhancer deficient mouse, where they specifically deleted a enhancer of this locus that is expressed in non-classical monocytes. And when you do this, these mice cannot develop non-classical monocytes, but it spares other defects associated with NR77 depletion that often will impact T cell function. So we want to take these mice and see whether or not there is a specific role for non-classical monocytes in BALL survival. And finally, we have patient samples from responders and non-responders of ALL, where we're looking to quantify the monocyte subset abundance versus patient outcome. At the moment, we've broadly looked at monocytes as a whole, but can we just break this down into different monocyte subsets? So that is part one of the talk, but I also want to show some unpublished data, which focuses on understanding how the surface antigen CD19 is regulated in human BALL. And this actually stemmed from working with a student in the lab, Eric Wang, who was screening for cell surface protein regulators in AML. And I thought, well, I can apply the exact same strategy to BALL, particularly with the emergence of CAR T and immune-based therapies. And this was further inspired by chats with Eric Kohler in Atlanta, where he really suggested how important antigen abundance is for T cell responsiveness. And we know here that the interaction between immune-based therapies and BALL blasts is very complex. Not only does the CAR interact with its target based on the antigen, but there's many microenvironmental factors that may impact the disease course. But just focusing on the antigen abundance itself, many studies of clinical trials using CAR T have shown the emergence of CD19 negative relapses of BALL following CAR T cell therapy. And this may correspond to CAR T cell persistence or the presence of pre-existing CD19 negative populations, for example. But not every case of CAR T cell reemergence can be explained by deletion at the CD19 locus. There's a lot of evidence out there now for splice isoforms of CD19 that can evade CAR T cell recognition. Phenomena like trogocytosis, where the target of the CAR is literally ripped off the surface of the cell, which attenuates the ability for a CAR to actually recognize its target anymore and kill. And antigen masking, which was a production error leading to masking of the antigen on the surface of BALL, disabling the killing capacity of CAR. And work from Terry Fry's group has actually shown quite clearly that the amount of surface antigen on a BALL blast is very important for driving a CAR T cell response. And using an engineered form of NALM6 BALL cells, they were able to show that in BALLs that had high expression of CD22 on the surface of the blast, these cells were able to induce a greater expansion of CAR T cells following infusion when compared to those cells that lacked expression of CD22. Furthermore, patients that achieved a MRD negative or not MRD negative remission following CD22 CAR induction could be fractioned based on the expression of CD22 density. And that's quite an important point, suggesting here that a lower density led to a lack of complete clearance of the tumor at the time of MRD quantification. But stepping back from just the CAR interaction with the CD19 and the surface antigen, we wanted to understand, well, can we identify the very basic mechanisms that may underpin the target antigen expression on the surface of the cell? So to do this, we used a CRISPR-based screening approach where we had Cas9 competent BALL cell lines. I focused on 3 cell lines here, NALM6, 697, and REH, which all have natural differences in surface CD19 expression. I transduced each cell line with a genome-wide sgRNA library, allowed cells to edit for 12 days, and then transduced cells were separated on the basis of either high CD19 surface expression or low surface CD19 surface expression. And we compared sgRNAs that are enriched in these 2 different CD19 expressing BALL populations. So the data actually looks like this. On the y-axis, we have the CD19 score. So the higher you go, these guide RNAs are enriched in CD19 high BALL blasts. And the lower you go here on the axis, these guides are enriched on the CD19 low BALL blasts. And what we saw first and foremost was our top hit was CD19 itself. So that's great news. It means it worked. And we were very happy to see that. CD81, which is critical for the maturation of CD19 and transiting to the cell surface for stabilization, was also a top activator of this protein. And PAX5, which in the 1990s, Meinrad Bosslinger and others were able to show that CD19 specifically binds the promoter of PAX5 specifically binds the CD19 promoter to activate gene expression. So we then wanted to look across our cell lines at common mechanisms by which, when you hit a particular target gene, CD19 levels drop. Can we identify conserved mechanisms that would regulate antigen expression and potentially represent mechanisms of resistance in CAR T cell therapy? So the design of this experiment was to take REH-Cas9 cell lines and infect with a cherry-based sgRNA vector. And I've shown here a non-targeting control and CD19 versus mCherry. What we then did was we allowed editing for 1 week using guide RNAs, and we quantified the mean fluorescence intensity of CD19 in sgRNA expressing cells, subtracting away the mean fluorescence intensity of the non-expressing cells. So what you can see here is the difference in non-targeting controls is negligible. We don't see any difference in guide expressing or not at CD19 level. However, if you hit CD19 itself, now we start to see a drop in surface antigen expression. But most strikingly, the top hit in our screen was this gene ZNF143, which I'll now go into a bit further. This is just representative gating of the CD19 expression in vitro, where we see here that CD19 guides seem to produce, compared to our non-targeting control, a heterozygote-like state and homozygote-like loss of 19, whereas ZNF143 is more of a uniform downregulation of CD19 following editing. To test whether or not this is actually a significant level of CD19 downregulation, we also generated, using a lentiviral vector expressing a CD19 CAR with a 41BB CD3 zeta construct here, we took human CD8 T cells, infected these primary T cells with the CAR, and then challenged pools of cells that had a subpopulation expressing the sgRNA. And what we did was that we either used empty CAR T cells or transduced to challenge these pools, and then we quantified the representation of guides after challenge. So if they're positively selected for by CAR, we would expect a higher proportion of mCherry positive cells compared to empty T cell challenge populations. These CARs in the preliminary studies that I'm presenting showed using this effector to target ratio, showed a dose-dependent response to BALL in vitro. And if we focus in on the raw data of these particular samples, we can see that in sgRNA ROSA cases, so a non-targeting control, the proportion of mCherry positive cells following CAR challenge is comparable. However, with ZNF143, in empty T cell challenge cells, we can see here CD19 levels are much lower than in the empty ROSA control, and these cells are actively selected for by CAR T cells based on their lower antigen expression. And we've quantified that here, not reaching statistical significance due to largely a lack of replication at this point, but it's looking like they do drive resistance to CAR in vitro. So how does ZNF143 actually function normally, and how can we explain how ZNF143 may regulate CD19? And to do this, we really went back to the literature of ZNF143 and how it functions across the genome. And in ChIP-seq studies published in 2015, across 3 different cell lines, they used K562, a GM B cell line, as well as a hepatocytic line. They were able to show that zinc finger 143 binding was enriched at the promoters of genes with POL2. However, there was some co-localization of ZNF143 at enhancer and insulator elements characterized by cohesin and CTCF binding. So this suggested to us that ZNF143 may represent an important factor in not only driving transcription from promoters, but also potentially driving the formation of structural loops that form between promoter elements and enhancers. And was this the mechanism by which it was defining CD19 expression? So I'm just showing some raw data here with 3C studies demonstrating the binding of ZNF143 at the promoter of these cells corresponds with looping events with upstream enhancers of this particular gene. And when they use siRNAs to knock down ZNF143, we saw a loss in these looping events. So we want to test whether or not, first, ZNF143 regulated the looping of the genome that may regulate downstream gene activity. So to do this, we used a technique known as Hi-C, which is a proximity ligation-based technique whereby you take your cell line and you cross-link DNA-bound proteins to DNA, and then what you will do is, following cross-linking, just randomly chop up the DNA that's bound by protein using Hin3. Then what we do after that is we fill in the ends of these cuts with a biotinylated mark and perform a blunt end proximity ligation. And what this gives us here is the ability to pull down strands of DNA that were in proximity to one another in three-dimensional space through DNA sequencing subsequently. And the data looks like this. So on the diagonal, we see this is the linear genome on the diagonal. And if we take this corner of topologically associated domain 1 and this corner here and extend to the diagonal, the intensity of red here corresponds to the intensity of interaction that occurs between these two distal regions of the genome in three-dimensional space. So what we thought here was, well, do we see different activity levels of genome topology in CD19 expressing cells or CD19 deficient cells? So now looking at a CD19 expressing cell line GM12878 or CD19 negative MOLM13, I've highlighted a topological domain that contains CD19 itself. And as you'll see here, the intensity of DNA interactions in this region is significantly higher than in cell types that do not express CD19. But are these the result of ZNF143 establishing a greater level of interactions at this particular locus? So to look at this, we performed Hi-C analysis following knockout of ZNF143 compared to SG-ROSA in our BALL cell lines. And quite surprisingly, we actually saw no real global differences in the genome chromatin structure following ZNF143 knockout. Indeed, when we quantified changes in the mean TAD activity, so those are those topological domain, the amount of DNA interactions occurring within each domain, comparing our SG-ROSA and ZNF143 guides, we saw almost no difference of statistical significance between any cell lines comparing these two conditions, suggesting here that if you knock out ZNF143, there seems to be maybe a subtle loss of interaction, but nothing really that important for establishing the architecture of chromosomes in a BALL cell. So next, we then zeroed in on the promoter itself using this data. And what you can actually do here is look at a specific sequence of the DNA and then quantify the amount of times that that sequence of DNA interacts with a distal enhancer or other DNA element. And this is a technique known as virtual 4C, where, for example, we took the CD19 promoter and we looked for hybrid reads that were located away from this promoter to determine whether or not there was a looping event happening. And what we found here is the majority of DNA interactions of the CD19 promoter occurred immediately proximal to the promoter itself, suggesting here that this gene probably doesn't have tissue-specific enhancer elements. And that was highlighted further by H3K27 acetylation and RNA sequencing, where we see here, for example, this peak in H3K27 acetylation at this ATXN2L gene, corresponding with transcript activity from this particular gene. And although we may see some interaction between these regions, it's probably quite negligible, as this is a very active genomic region. So we think here that the CD19 promoter is doing the lion's share of the work in regulating CD19 expression. So to delve into this further, we then took ChIP-seq data from ZNF143 in CD19 expressing lines or CD19 negative lines. And we're able to see here that in CD19 expressing lines, we saw a beautiful peak of ZNF143 right on the promoter of this gene. Furthermore, when we just knock out ZNF143 across multiple cell lines down here on the right side compared to SG-ROSA of these three independent lines, the vast majority of differentially expressed genes were downregulated upon ZNF143 knockout. And what that suggests to us is the majority of the activity of this gene may be involved in transcriptional activation. And using GO term analysis, we see that ZNF143 knockout appears to drive a loss of genes involved in mRNA processing when compared to CD19 or non-targeting controls, suggesting we're getting really a shutdown in transcriptional activation following knockout of this gene. So to conclude the second part of this talk, and just to wrap up, we were able to use CRISPR to identify regulators of CD19, and we found this novel regulator, ZNF143, appearing to positively regulate CD19 expression. But we don't believe this is through altering chromatin topology. In fact, we do seem to think that this is due to direct binding to the CD19 promoter, and we want to now identify interacting partners such as POL2 and PAX5, which may be critical for ZNF143's activity at the CD19 promoter. But we also believe that ZNF143 is a more broad transcriptional activator. It really doesn't seem to have a CD19 specific function. But one of the more interesting points of our screen, and something that we're looking into now, are these sgRNAs that cause an upregulation of antigen on the surface of BALL. And this is really important because if we can increase antigen abundance on BALL blasts, similar to how Terry Fry's group used bryastatin-1 to increase CD22 antigen abundance on BALL cells, we might be able to increase the sensitivity of CAR to its target. So just to conclude, really the first part of the talk highlighted the approaches we use to look at microenvironmental interactions with leukemia. And I believe now that there's an opportunity to explore how the environment evolves during those really immediate stages of CAR T-cell infusion. We already know that monocytes and macrophages are critical for driving a cytokine release syndrome. However, what are the particular players in the microenvironment that may potentially hinder CAR activity and or promote BALL survival? And then finally, using our screening approaches, we believe we've identified a nice strategy to look at targets that may be involved in antigen escape during BALL therapy and predict some of these potential mechanisms. Is ZNF143 dynamically regulated in some cases to drive a drop in CD19? And also, can we target antigen regulation to increase the abundance of antigen and CD19 therapeutic sensitivity? So with that, I believe I am definitely out of time, and I want to thank Yanis Ifantis for all of his support throughout this postdoc, as well as Igor Dolgolev, who is the co-first author on the first part of the talk. He's a wonderful bioinformatician and person. Bill Carroll has been really amazing in clinical insights into BALL and introducing me to the Children's Oncology Group, which has been such a welcoming community. Chao Ma and Eric Wang are two really phenomenal students that helped with the microfluidics device and CRISPR screen respectively. Karen Rabin, Charles Mulligan, and Eric, all from the COG and St. Jude based meetings. Eric in particular for his discussion on CD19 antigen abundance that drove a lot of the second part of the talk. And I'll happily take any questions.
Fantastic, thank you, Matt. Okay, so there is one question as people straighten out all of the information you just overwhelmed them with. But let me ask something that's on the top of my mind, and I'll move to that first question. You talked all about BALL, but I wondered if you've addressed the recruitment of non-classical monocytes in a AML situation too, or if you think the AML itself provides some of those cues.
Actually, work from Jeff Tyner's lab pretty much identified these cell types in AML, I believe. Not to the extent they basically said there's a CSF1R non-transformed population that coexists with their AML and does provide some protective function. So I believe it's been identified in AML previously. The difficulty there is you do need this kind of Peter Van Galen style detection of AML-associated mutations to separate the transformed and non-transformed counterparts truly from these individuals and determine what is actually leukemia-associated or leukemia itself. BALL is super easy. You just, they're B cells. They sit over there.
James DeGregory has a question, which I'll let him ask.
Hi, great talk. So back to the first part of the talk. So I'm sure you know Mel Greaves has proposed this delayed infection model for BALL risk. And and I don't know, I'm wondering what your thoughts on how that might tie in with what you're showing. In other words, how might the infection associated changes in, the immune milieu, including a lot of the myeloid compartments, influence this risk, and how might it tie in with what you're showing with that M1 compartment?
Absolutely, yeah, it's a really good question. So it's so hard to with the infection-based hypothesis, there's a few issues that arise from that. I mean, these are largely correlative studies that occur when you have like an outbreak of influenza and they see more ALL cases and so forth. We don't actually know the makeup of those patients prior to emerging ALL. So sure, absolutely, maybe they have a shift in the polarity of monocytic populations during that acute infection. However, it's really hard to understand the impact of that on tumor progression unless you model that specifically. Another thing regarding that Malgreave studies is the emergence of oncogenic lesions in utero, which I think is extremely interesting. How does that in the hematopoietic stem and progenitor compartment lead to altered function of these immune-based interacting cells later on at the development of disease? We don't really know. I mean, BCR-ABL is kind of a really nice avenue to go down here, where you can get switches in lineage carrying the same oncogenic lesion. How does the presence of BCR-ABL in a myeloid CML-like disease alter the function of a more mature monocyte-like cell that it may be interacting with the blast population? And then when it switches to ALL, what happens to those cells? And in fact, sorry, this is the longest answer. I really like this question because one of the cases that we describe in the paper is a BCR-ABL positive case. The patient arrives with B-ALL, and this patient had the non-classical monocyte swing in population. They then relapsed with an AML around 4 years later in our study. Well, they described it as an AML, the pathologist, but it was a CD34 positive, 19 negative disease. And the same myeloid skewing existed in those patients. So long story short, I think that this response is just an inflammatory response,
right?
Largely. And it's really not a leukemic specific response in that case. So maybe the hygiene thing's real, but it's next to impossible to test, I think, his studies.
Yeah, you may get some of the same cytokines. I know IL-6 is produced by these BCR-ABL positive cells, and so that's certainly a good candidate.
Absolutely, yeah. I think BCR-ABL is a great way to go with this. Very interesting.
Yeah, thanks.
So the next couple questions in a minute or so is Mike Verneris. Mike, you want to go ahead, and then Eric Kohler.
So Matt, great talk, really interesting. So the nomenclature on, I have two very quick questions. The nomenclature on monocytes is a bit confusing to me. So are these alternatively, these alternative monocytes, are they basically like the ones that are in obesity and are inflammatory? So that's the first question. The second question is, have you ever, as you know, pertinent to this conversation you're just having with James, as you know, people with CML, are kind of relegated to lifelong tyrosine kinase inhibitor therapy. And there's been this movement to withdraw TKIs in a small fraction of people, and about a quarter, 20% or so, you can successfully do that. Now, have you ever considered looking at your bone marrow monocytes as a predictor for whether or not you can take away TKIs? So I'll let you answer both questions.
That's a great idea. So whether or not the abundance prior to TKI withdrawal does appear to coincide with relapse, it's a great question. We haven't actually thought about that at all. Those large cohorts where they're doing the withdrawal, I believe they're in Adelaide in Australia with Timothy Hughes. They did that study. So it would be interesting to ask them whether or not they have quantified by flow cytometry different subtypes of myeloid cells, when just serendipitously, if they were looking for CML blasts at the time of withdrawal, if they have information on the normal BCR-ABL negative cells at the point of withdrawal. So that's a really interesting point. To the first point, which is the association with inflammatory diseases. So that point is important. So these are the monocytes that are associated with atherosclerosis largely, and that is work from many labs, including the Hedrick lab, where they have used a series of mouse models to show that the progression of atherosclerotic plaques depends on the presence of these monocyte-like cells. However, we did not see a clear pro-inflammatory signature in these cell types in our data sets. Most of the changes in gene expression we saw were largely down to things involved in migration, which is a major function of these cell types, is to migrate along endothelial cells. But pro-inflammatory like cells, we didn't make that conclusion based on the cells that we saw. It was a stretch. So we don't think they're promoting a pro-inflammatory environment. They may just be involved in repairing the consequence of an inflammatory environment.
Great.
So one last question from Eric. We're a little bit over, but I'll beg for forgiveness from the student participants after this.
Hey, Matt, really nice.
Hey, Eric.
Your ZNF 143 story, it's really interesting. Have you looked, or is there any way from your screen that you can see if it is altering splicing of the CD19 gene? Because alternative splicing is probably a mechanism of some CD19 escape in CAR T cell treatment.
Absolutely. Yeah, I think actually the other way, CD19 repressors actually appear to have a lot of splicing regulators in there, novel splicing regulators that we're still trying to figure out what they do. But yeah, splicing has become a major leader in the candidates for when you hit these targets, you have an upregulation in CD19. Are we getting a loss of genes that otherwise destabilize CD19 splice isoforms, or we're not sure. But ZNF143 canonically is thought to be just a transcriptional activator. I'm not sure about, there's no alternate promoters of CD19 that it seems to bind. So at least the transcriptional start site is consistent, and I'm not sure how much of an impact it has on splice isoforms that are then matured in the cell. But really, the literature at this point would suggest it may be just like an on and off switch, as opposed to other targets we identified that may be involved in the full complement of full-length CD19 maturation.
Cool. Thank you very much, Matt. And I want to thank everybody for their questions and attending. So now if everyone else wants to leave, the lunch afterwards is for students and fellows. So
thank you very much.
I'm just going to hang out until that gets underway. So I hope you can answer some more questions there. We have, I think, who is in your group here is Laurel, who's an MD PhD student working on solid tumors, head and neck cancers, and is interested in immune infiltrates.
Great.
There is Taylor, who's a immunology student with Eric Pietras.
Dallas Jones.
Dallas is in
right now.
yeah, so you guys can all, open up your screens, whatever. So Dallas is in Mike Verneris's lab as a fellow. Who else is coming? Annika had to bail because she's doing a mouse experiment right now.
Classic Annika.
Huh?
How dare she?
Classic Annika.
Yeah, I can't believe what's happening.
So she would be the Craig Jordan representative, but she's not here. And who else do we have? I guess we have some other volunteers maybe still on the line.
Okay.
But basically what I thought you guys could do is tell Matt like what your project is, what you're interested in. And of course, if you have any additional questions or anything you want to ask, now's your time, and I'm going to leave.
Thank you, Patricia. Cheers.
No problem. See you guys.
Thanks, Patricia.
All right, so we have a few people here. We have
you have some extras. I can't explain that, but
that's great.
So there's
do whatever you want with that.
Yeah, so there's Maria Amaya is here as well. So she's from Craig Jordan's lab as well. She's a fellow, a medical fellow. So
what she was working in the tissue culture hood with Annika, so they may just try to listen in together. So,
probably. What I wouldn't be surprised.
I'll ask them questions. See what they do.
I can also text them and just like give them a hard time the whole time.
Good.
Okay.
That's how we do
Okay,
it.
but yeah, so if you guys would just sort of introduce your projects, that would be cool.
Awesome. Thank you very much, Patricia. Awesome. Thank you for hanging around. my voice hurts. So
all right, well, we'll give you a break, I guess, for a minute.
Thanks, mate.
I will.
I appreciate that. Yeah. Give
Yeah,
us a
no,
go, Taylor. What do you got?
You went an hour straight. I want another half hour out of you. Come on. So yeah, I mean, obviously super cool talk. I'm sure you don't need to know that. But I really appreciated the use of like just some really basic, functional phenotypic assays within like a really high level site-seq, high-C, all those types of things. So I thought that was a really great way to bring those in, because usually it's like kind of one or the other. I noticed a lot in papers. So I really
I agree. We were getting stung by reviewers in the lab because we just kept sequencing stuff and then being like, oh, it's biology. That's not true. I mean, you can't do that. I could just sequence anyone off the street and tell them that this is how science works. But preclinical platforms are so important. My old boss in Australia, he was obsessed with it. He would tell me all the time, like, you don't publish anything without a preclinical part, and he was so strict on it. And I think it was really good training. And then with Giannis, we had a lot of people around that could help us out, because collaboration's the only way these things get done in any project in all science.
Yeah, definitely. So
yeah,
my stuff's very different, but I'm definitely really interested in immuno-oncology is where I want to be going. But as Patricia mentioned, I'm in the lab of Eric Petrus, or just traditional hematopoiesis and inflammatory hematopoiesis, looking at his seminal paper being chronic IL-1. And so I've worked a lot with chronic autoimmune disorders, looking at lupus and collagen-induced arthritis and how those affect the hematopoietic environment and stuff. And then now my main focus is actually looking at how metabolites and metabolic shifts post-immune activation affects the ability of HSC and progenitors to then be further activated, like in a single immune response kind of deal.
So is this using models like TET2, DNMT3A?
So mine is much more basic, just standard, just wild type system, but I am actually starting to look into the TET2 system.
Yep.
And I'm actually more interested in like the idea of what the field isn't looking at. Everybody looks at an acute or a chronic. And I'm interested in like the middle ground, which is on multiple acutes with like a washout period. And so
we're humans. I mean, that's what we experience.
Right.
So that's really important, I think. Our lab did a lot of work on clonal hematopoiesis using TET2 based models.
Yeah, exactly.
But really didn't address. I mean, Eric gave a great talk at ISCH about chronic inflammation.
Yeah, that talk, exactly.
Yeah, so I think it's a great way to go, especially in terms of how these CHIP patients do emerge. And the main phenotype they have is cardiovascular disease. It's not AML.
Right. Yeah. And also how like that all ties into the trained immunity and the trained immunity literature. And like, that's kind of where I've tied it all kind of integrated it all in this like metabolic analysis of HSC and hematopoiesis, and looking at like things like itaconate that comes out of an alternative metabolite out of the TCA cycle, and how that can kind of adjust the responsiveness of cells and stuff like that, and how they can, alter their phenotype just simply because of CHIPs and metabolism.
Do you ever, like, we would always go back to the methyl cellulose assay.
Yeah.
Don Metcalf was one of the first people I ever interacted with in my PhD, and he kind of made that a big, beautiful tool for hematopoiesis. Do you ever use these oncometabolites in these colony formation assays and really purify your stem cells and then hit them with the metabolite?
That's like, that's like an immediate next step experiment. Actually, I'm going to be doing here in the next week or two.
Awesome. That's going to be a good one.
Yeah, exactly. Looking at that, and as well as just like simple liquid culture and stuff. But yeah, we will be moving this kind of into the TET2 landscape, and obviously these metabolites, succinate, fumarate, and stuff, they all have a huge role in the deoxygenases and whatnot. So so that's kind of like how I'm going to shift my project on like basic hematopoiesis and immune stimulation into kind of like the onco-immuno and metabolo kind of field.
That's very beautiful. Laurel, I believe you're there.
Yes.
Right. Hey, how you going?
Thank you so much for the talk. That was great. I'm slightly different, so solid tumors, adults, head and neck cancer, actually in a radiation oncology lab, but I'm studying right now FRNB2 knockouts on the vasculature and how that affects the tumor microenvironment and systemic immune environment surrounding that. So yeah, this is a great talk about vascular stuff, of course.
We had this great radiation oncologist in the lab when I first joined. His name was Filmo.
Oh, cool.
And he was this dude from North Carolina, and he was one of the first people I met. I was when I got to America. I thought everyone had a gun. So he taught me that is not the case. But Filmo was working with these amazing head and neck radiation tools where you would actually have a tube that went through the orifice here, and there was like a radioactivatable probe that sat next to the tumor. Then you excite it, and it would kill the tumor. It was crazy. That guy was a maniac, but really beautiful work. So yeah, I like the vasculature stuff. I think a few labs at NYU have looked at solid tumors, not head and neck, but gliomas and so forth, using our fluidics devices. And it's been really fruitful for them in terms of modeling things like hypoxia, angiogenesis in general in those tumors. So do you look into stuff like that, making mimetics of the disease, like 3-dimensional mimetics?
We're not there yet. It's definitely something we want to do. I'm actually like in my first year of my PhD. So right now I'm kind of more just like describing the phenotype, and we're kind of moving on from there because we're not sure if it's like a transepithelial migration thing or an angiogenesis thing, or there's like survival proliferation of the cells. So we're not really sure how the B2 knockout right now is changing anything, but we know it is. So
we went through this whole phase of like single cell and everything, which everyone did. And part of it was the vascular niche of the bone marrow. And then subsequent to that, there were a series of studies where they took the vascular from all tissues and profiled them at that level. So I wonder if your vascular cells are in there and you can just look at natural patterns of expression of your gene in these existing data sets.
That would be great. What is that called?
If you just look for like that, it's always in Nature or something, where they just single cell stuff and they're like, we did it. Here's a heat map, aka my paper. So then what you can do is look into it and say, you know, if they have vasculature from that region of interest, maybe your gene is uniquely expressed in a subpopulation. That's how we kind of got into the single cell realm of vascular endothelium, and it was a lot of fun.
Yeah, that would be great, because actually one of the questions right now is we want to know if it's like changing, if it's more expressed on tumor vasculature specifically, and we do radiation. So we already showed it increases after radiation, but we don't know what it's compared to normal. So that would be a good data set to like.
Yeah, normal is like a great start. And if you can get some of those cells out, I mean, also spatial transcriptomics is all the rage now. And for things like vasculature, you can pick them up really nicely using those techniques. But once again, I don't think you ought to depend too much on single cell approaches either. I think you should go into, I mean, my study is a great example of that, but at the time we were really fishing for a result. What you've come in with is a hypothesis about a gene. I think that's a great approach. And then use the single cell as a tool to look for these expression patterns, but don't depend on it for your results, because it nearly killed me.
So we're trying to focus more on mechanistic studies to really narrow down what it's doing.
Yeah, it's beautiful. Well, good luck, and
thank you.
Congrats on getting into the PhD. Who we have? Thanks. I'm just going to go in order here. We got Jill. Jill? We don't have Jill. We do have Jill. Carolyn? It's just a lot of people here. Lawrence Chan.
There's a possibility that these are people that like put it on during the original Zoom talk and then just like left their laptop. I'm actually guilty of that recently. I was like, I was getting my seahorse ready, and then I just like left the talk on and I left to go do the seahorse run. And apparently my laptop was like talking to everyone, and it was like, hey Taylor, so what do you think about that talk? You know, with metabolism, that's your thing. And my lab mate was just sitting there like, do I like answer? Like, do I tell them? So I gotta have that feeling.
They just think you're really mysterious now. So that's good.
I'm not an oncologist. Just enjoy
Lawrence.
Yes.
All right. Wonderful.
How you doing?
What?
Lawrence, we got you. Goodbye, Lawrence. That's it. That was quick.
All right.
Dallas? It's just the three of us. Well, maybe what I could do is like I'm at this stage now where I'm thinking about leaving New York, right? Obviously, it's like it's a pretty intense place to live. And it'll be probably the end of next year, the following year, that I would actually leave the place. How has Colorado been in terms of supporting you through your like recruitment, through your life? I mean, this is a big deal, like being a postdoc, how people take care of each other as an institute, you know, mental health problems, like physical health problems. Do you feel supported as young like people in the institute in having this kind of engagement about your health and well-being?
Yeah, I mean, definitely. So are you going to do another postdoc? Or you're saying for faculty, right? I was like,
faculty, yeah. No,
okay, good. The way that you said it, I wasn't quite so sure.
I can't do that to myself.
Oh, I was gonna, yeah, come on. But yeah, I mean, so physical health, you'll find at least on the campus and in most of Colorado isn't really an issue, right? Like we're consistently the healthiest state in the nation over time and over again. Around where the medical campus is in the city of Aurora, it's like it was classically like the not good area of Denver. And like, so it's like gentrifying up and whatnot. So you see that actually around the medical campus. And because we're such a regional hospital, you will see that kind of thing. But as far as like faculty in school and like, you know, people on campus, there's really like none of that. But then mental health is a massive focus of the campus. I can't speak so much for, you know, professor level, but at least as a graduate student, there's a mountain of resources. We are given free mental health services through the university, through our insurance. There's a local health services that use our student insurance. And like even through this, the one on campus, there's multiple offices, they have different styles. So there's like a lot of that. There's even further, like I just got an email today about like a Zoom meeting kind of group discussion on mental health that's going to be recurring. And this one being about like mental health during COVID and all that kind of stuff. So yeah, I feel like our university takes it extremely, extremely seriously. And
that's really good.
Like you will notice that the mentality is definitely not that of the coasts. And, you know, obviously amazing science gets done, but it is definitely not the same way. And there's actually, it happens where faculty come from those bigger institutions on the coast and that type of lifestyle and work style, and they come here and they have a really, really difficult time because that's just not how we recruit. You know, even, you know, our immunology program, the program I'm in, students lead the recruitment, and we talk consistently like that's not how we do things here.
Sure.
You know,
yeah, it's good. I mean, it's a hard thing to get out of though for someone like me. I think I went from like 6 to 11 here. Really, it was. I've never been like a relaxed person. I won't even pretend. Like in Australia, I just seem like Larry David compared to all my friends. But but then when I got to New York, it just really brought that part out of me. This like kind of intensity that I do enjoy, but it's not sustainable either.
No, and not at all.
I think that's something that, like, I'm quite young, that I would like to move away from that kind of intensity. I feel like I've been trained nonstop for 11 years and haven't really had a chance to.
Scares me.
No. But like I'm just like, I'm 30 now. I mean, I'm not.
Well, that sucks. I'm just about to be 30, and you're about to be a professor.
No. Like, it's not like a Dookie Howser thing. I think what it is in Australia, your PhD takes 3 years.
What?
They don't want
masters.
It's called honors. It takes 1 year before that.
So do you do 4 years of traditional university, 1 year of masters, and then 3 years of PhD?
3, 1, 3. Yeah, so it's 7 total. So I was 17, finished undergraduate at 20, started PhD at 21. So it was quick, and I think.
Wow.
But it's kind of nice because it throws you into a lot of environments fast.
Mm-hmm.
You don't get kind of caught in this 8-year thing, which, you know, we've had people in the lab be there for so long, and it's like they all want to go to biotech after it. And I don't want to have students that feel like I'm trapping them as like some kind of cheap labor. You want to recruit people that are interested in the work you do, and also feel like they're comfortable to come to work and not be worked into the ground. So my challenge is going to be getting in there and being like, I'm used to everyone being here all night. I have to start to like turn that off and be more efficient. It doesn't mean that the work gets any worse. Working all night doesn't do anything. Actually.
No, yeah, you're less efficient. Exactly, yeah.
Yeah.
And I think that's a part of what like people here recognize is that if you like submit the paper 6 months later because you enjoyed the 2 years a little bit more and had a little bit more balance, like obviously that really doesn't matter. I mean, I will say obviously it's going to be tough during those first few years for you, right? You know, that the junior entry level before you get your first R1 or whatever. But yeah, I mean, that's essentially how people out here think.
Yeah, I think that's really nice. I mean, it's also a chance for my family to visit. They don't like New York at all. They live in Tasmania. It's cool. Like I like it. I was playing music here for the whole time and like doing all the fun stuff. But then I do need to go somewhere where my brothers and stuff would actually like it. They live in Tasmania, so it's like it's our Colorado.
Yeah, no, that's totally interesting.
Yeah, so it's not very yeah, I don't see it as like just a work move. I mean, the labs there are great. The people I know from the children's oncology group are so nice.
Oh, all of our immuno-oncology, yeah, it's insane here.
And are you both locals of Colorado?
I am.
No.
Okay, where are you from, Laurel?
I was originally from California, Northern California, and then I went to school in upstate New York, and now I'm here. So
at Cornell?
Yeah, Cornell.
Oh, great. Is that in the forest in Ithaca there?
Yep, in the forest.
Oh, that's really nice.
Harry Potter over here.
Yeah, it's like Lego.
As far as Colorado goes, a lot sunnier, less snow, but great skiing. I feel like when I East Coast, Cornell was very East Coast mentality. And then I came here and I kind of had to channel some of my workaholicness into outdoor activities, which I find was actually excellent. I love skiing and hiking now. So that's been good. I'd say, yeah, I don't know, the atmosphere is just better here, but I feel like I'm getting the same amount of work done and you adjust.
Yeah, I think like my main thing is recruitment, getting people to join up a lab where the guys just starting their lab. It's easier in places like here because you have so many universities in one area, but you do get eaten up by the huge labs that are around you. So obviously
that's not as big of an issue on our campus. I think because of like how we recruit not only new faculty, but also students, like the university is so on top of like cutting edge, so like all the younger faculty we're recruiting are, Eric, for example, Eric Petrus, my mentor, like I was his first grad student. Like it was a year into him and it was really exciting because I wanted a mentor that would be able to be hands-on with me while being hands-off. Like I didn't need to be taught, but I wanted to have that like close personal relationship like you get with a younger PI that isn't off at a million meetings and all the time, kind of like us, right? Like I'm sure for grad students in his lab, it's not like they can have this general daily kind of interaction with him.
It's interesting. We present that kind of mentoring point as postdocs. So like I'm one of the older postdocs in the lab by experience now. So then what happens is you cluster them around each other and then you say like, what's going on? They won't ask questions, for example, at lab meetings. So I brought them into a room before COVID and said like, are you all right? Like, is everything, do you feel scared about asking questions? Do you feel like you're getting spoken over? Like, how do you, like things like this? And I mean, that's been a really good experience for me because I had 2 students. So it's not like a new thing to train students. It's more understanding like where they're going to come from in Colorado. We have someone from Aurora actually in our lab who grew up there. And but she's Ethiopian. Her family were there and they got kind of pushed out by the gentrifying of Aurora.
Yep. It's the really sad thing that's happening on campus right